is Dr. Caray Walker, so welcome back. Caray is a senior lecturer at microbiology at Anglia Ruskin University in Cambridge. Current research includes One Health approach to understand drivers of AMR and development of novel therapeutics, multidrug resistant pathogens, particularly bacterial zoonoses. So over to you.
Yes, I'm going to talk today about some work that we've just had published recently on the prevalence of multidrug resistant Pseudomonas species from wild bird fecal samples. But more so, I wanted to sort of talk about the fact that we do a lot of surveillance work essentially in the environment, and the importance of surveillance and finding bacteria that are resistant to antimicrobials and doing more research onto those. Using genomics which hopefully will help inform others on the types of mechanisms of resistance as well. So surveillance is one of the cornerstones of infectious disease management, which is mentioned in the O'Neill report. And the important thing for us is to look at the reservoirs of resistance. So we want to know what types of resistance mechanisms are circulating in the environment, and particularly looking at the emergence and spread of resistance to some of the newer antibiotics, because a lot of the surveillance studies so far don't tend to look so much at the newer antibiotics. So looking at the mechanisms of resistance that are circulating locally, that's the work that we've published recently, but we're also looking to do that on a more national scale, and also in collaboration with some other groups in other countries as well. Poland, we're having chats with some people there that are doing some work. So the main thing is really then to have an idea of the resistance trends. So to see how resistance mechanisms are potentially changing over time, and with seasons as well, particularly when you're talking about wildlife, that move around, migratory birds, that type of thing. And we are taking some of these, or quite a lot of the samples, and using whole genome sequencing to identify the mechanisms of resistance and to look at changes in those. Obviously, surveillance is also really important to inform public health policy, legislation, and infection control plans as well. So another extra thing. So how does surveillance help with therapeutics? As I mentioned, we use whole genome sequencing data, or use whole genome sequencing, sorry, to look at some of our more interesting isolates that we identify. Particularly if they're multi-drug resistant or extremely drug resistant, we are taking those further to look at, to get whole genome sequencing done. We are also going to be doing some whole metagenome sequencing as well at some point. But particularly we're looking at single nucleotide polymorphisms, SNPs, to be able to understand the mechanisms of resistance and how those change. Are they changing over time? And then also to look at the transmission potentially of these. So by keeping the surveillance kind of going, you can work out whether these mechanisms, if they're changing, are they then being transmitted as well, and how far afield is that happening? Hopefully this data then will provide insights into design and development of new therapeutics. And when I mentioned therapeutics, obviously we're talking about new antimicrobials here a lot today, but also the other important thing for antimicrobial, to prevent antimicrobial resistance, is to have good diagnostics as well. So that's something that this work will also help with, hopefully, is to inform for diagnostic tests, for example. So as I mentioned, we're mostly involved in wildlife surveillance and environmental surveillance. So it's obviously essential to look at pathogen reservoirs that could be in wildlife, and obviously this is quite apt, I suppose, for thinking about spillover events and talking about coronavirus. So we're really, as part of that, and obviously there are some large surveillance that goes on, but what we have found is that there is not a huge amount of surveillance that's happening looking at antimicrobial resistance and antimicrobial resistance genes in wildlife and in their particular sort of ecosystem environments. And obviously looking for this type of resistant bacteria in wildlife can be a sign that or can give us some indications to the drivers of antimicrobial resistance as well. So particularly thinking about human activities, effluent that's going into rivers, that type of thing, slurry, and also in other countries as well, thinking about manufacturing waste, not quite so much in the UK, but in some other countries where there's a lot of antimicrobial manufacturing sites that have waste going into water. So our work that I'm going to talk to you about in a minute is based on wild birds, and the reason we were most interested in those is because obviously their mobility, they can travel over long distances and poo a lot. So they may act as vectors for resistant bacteria because they frequent obviously around rivers as well, a lot of waterfowl that fly long distances, they hang around in water, and if that water happens to be receiving any sewage effluent, this is the type of thing that we were sort of we are interested in. And then obviously birds then move around and could potentially contaminate somewhere else as well. So more than 60% of human infections each year are attributable to zoonotic diseases, and currently there's quite a limited understanding of the natural prevalence and circulation of resistant bacteria in the wild animal population, and particularly in birds. And the reason for this is that a lot of the studies that are done look for diseased animals, and as part of that, the AMR is kind of done alongside this. But what we would like to see is a more national kind of surveillance of wildlife and taking a One Health approach. So looking at, river, soil, birds, other small mammals that might be frequenting in areas, and trying to see if there's any sort of links between those using genomics. And as I say, it's currently limited at the moment. There are studies looking at things like bird flu, for example, but again, not specifically looking for resistant bacteria. So our study was, it was over a 2-year period. 115 swabs. This was a fairly small study. This is one of the first ones that we kind of started. It was the 10-mile stretch around the River Cam. The important point there is that we tried to pick areas where there are a lot of human interaction, so people hanging about, boat clubs, nature reserves, that kind of thing. So that was kind of our reason, really. We wanted to see, could this potentially be a transmission point? We found, I guess unsurprisingly, that because obviously Pseudomonas are ubiquitous in the environment, but we found 21% of our samples had Pseudomonas. Said potentially dangerous because all of the Pseudomonas species that we found were resistant to at least one of the drugs mentioned here. And 75% of them were multidrug resistant to these drugs. So that was quite a sort of a sobering percentage, I think, to see that. We were a little surprised. So this is just a map here showing where the coordinates were. But the interesting point here is that the resistance we found was widespread. There was no statistical significance between where these isolates were found and the type of resistance, or the amount of resistance. But I guess the fact that they were bird fecal samples, that isn't unsurprising because obviously birds are mobile. To find more direct links to the particular area, we would probably look at a mammal maybe that doesn't move around so much, or look at soil samples and things like that, which again we're hoping to do. So we're unable to say obviously where the birds picked up these resistant bacteria or where they may transfer it to. And again, I think the important thing is here that further work is needed to understand our birds' potential vectors, bringing these resistant bacteria from other places when they're migrating. So it's something that we want to try and get some collaborations with people to find that out. And yeah, we made the news a bit with this, as you can imagine. Deadly bug, which is a bit interesting. So we went on from that, and we picked particularly the multidrug resistant isolates. I'm showing you the data from, or some of the data from the Pseudomonas aeruginosa isolate that we found. We had quite a mixture of different ones. There was only one actually aeruginosa. The rest were kind of environmental isolates that you would kind of expect. But interestingly and quite pleasing, the phenotypic data matched the genotypic data, so we were happy about that. And obviously we know that Pseudomonas do show a high level of intrinsic resistance to antimicrobial drugs. That is kind of expected. But I think this also makes it more important as well, because we know that they have the ability to become resistant very easily, mutations in chromosome, and obviously mobile genetic elements as well moving around. So the important thing is that, you know, these bacteria are present. This was just a random surveillance, nothing to do with any diseased animals, and we found a lot of resistance. So that is slightly worrying. So yeah, very short and sweet. We're hoping to continue this work. We've also got some collaborations with some wildlife centres around the UK as well. So we're hoping to continue to get some samples from them as well. And we're going to use the genomic data to look at circulating resistance mechanisms and changes over time. We've got a longitudinal study going on at the moment to hopefully again inform new therapeutics. Also in collaboration with Professor Melody Clark at BAS, we're looking to understand more about the anthropogenic drivers of AMR by looking at doing surveillance work in Antarctica, which is very exciting. So yeah, we're hopefully using some of this data to, as you've seen earlier, Chisholm and Alazar also in this flash talk, we are looking at developing new therapeutics. And there's also Plamina, who's looking at monoclonal antibodies as well. So yeah, that's it. So thanks to everybody. Is there any questions?